right. Good morning, everyone. Thank you for the kind introduction. Thank you to the organizers for their invitation. I'm really excited to be here to talk about some interesting work that we've been doing in our lab around deep learning modeling of genomics data. Initially, I had submitted spatial and temporal modeling of single-cell data sets as my title. But given the time frame, I think I'll only be able to fit the spatial part, and then I'm happy to talk about the temporal modeling part offline. And by spatial modeling, I mean the 3D organization of the DNA, which would be a nice primer for the next talk in this session. All right, so I don't think this audience needs that much motivation, but DNA is the basic building block of most organisms. The DNA gets converted to RNA, which forms the proteins. Proteins are the structural components of our body. And this flow of information going from DNA to protein is controlled by a multitude of factors that have to come together at the right time to ensure that the cell develops properly. And a lot of things can go wrong along this way that can lead to a wide variety of diseases. And so in order to try and understand the DNA, we sequenced it almost 2 decades back. And what we have realized since then is that just looking at the DNA sequence will not give us all the answers we are looking for, because while all the cells in my body are effectively have the same DNA sequence, they all are expressed differently. And that's because the genes that regulate these different cells are expressed differently. And the regulation of gene expression is composed of a complex machinery, which works like this beautiful symphony that comes together for cell regulation. So starting off with gene expression, certain genes need to be expressed at a certain time. This, in turn, is controlled by the binding of the transcription factor protein that signals the start of the gene transcription, which in turn could be affected or controlled by different types of chemical modifications happening on the DNA or in the environment of the DNA, which in turn could also be controlled or affected by the chromatin accessibility. So the idea being, if the DNA is open and accessible, then things can come and interact with it and affect the gene expression, and if not, then not. And all of this kind of nicely comes together in the way that the DNA is arranged in the 3D space inside the nucleus, which can control all of these different things. So luckily, or thankfully, we are able to sequence all these different genomic phenomena. So we can measure the gene expression, we can measure the TF binding, the modifications, the accessibility, and last but not the least, we can also measure this arrangement of the DNA in the 3D space. And with the advent of all these different technologies, what a lot of researchers are now interested in is multi-omic studies. So the idea here is that we want to jointly study all these different types of sequencing data sets together so that we can get a comprehensive picture of cell development and diseases. And one of the things that we are really interested in bringing in this multi-omic study is the spatial arrangement of the DNA. So when you think of DNA, for most part, you think of it as a linear entity, and that's totally fine. But the 3D arrangement of the DNA is also very critical to try and understand gene regulation. And that's because an enhancer, or a part of the DNA that's really far away from the gene in the linear space, could actually be interacting with it or affecting it because the way the DNA loops around in the 3D space. And researchers have found evidence that this is important for gene regulation, and this has also been seen to affect gene misregulation, resulting in disease states. So given the DNA organization, like I said, we are able to measure it using different types of sequencing technologies. The one that is very popular, it's known as Hi-C. And the output that we get from a Hi-C experiment is basically this matrix where the rows and the columns are the genomic regions. And if 2 regions are interacting frequently, that represents the intensity of the color. So it's some real signal value. And what we and some of the other researchers have done is that we have modeled this Hi-C data as a graph. So here in my graph, each node is a genomic region. the rows and the columns from the matrix. And then if we have evidence of two genomic regions interacting, we connect them in this graph using an edge. And the reason we do that is because now this modeling of Hi-C as a graph allows us to do some very interesting deep learning through the form of graph neural networks. So on a very high level, if you are familiar with convolutional neural networks, which are very popular for images, or well, used to be, the main idea behind convolution was that you learn a representation of a pixel in an image by taking a weighted aggregation of its nearby pixels. With graph neural networks, you basically perform the same operation. There are many formulations, but they all roughly do the same thing, which is we do a graph convolution where we learn a representation of a node by taking a weighted aggregation of all the neighboring nodes. So the input into my graph neural network is basically twofold. First is the set of node features, and the second is the adjacency matrix. So going back to our genomic graph, my node feature could be any genomic measurement that I've taken for that genomic region. It could be a ChIP-seq profile, it could be ATAC-seq, whatever is relevant for your task. And an adjacency matrix for this graph looks something like this, where all the nodes are arranged in rows and columns, and if two nodes are interacting, we give it a value 1, and if not, 0. Now, this is a very simplified binary representation, but if you have a weighted graph, you can imagine this being real values. So having done this modeling with bulk Hi-C data, we started looking into single-cell data sets. So the idea behind bulk measurements is we take a population of cell, we grind it up, and we get some sort of an average signal from the cell population. But what single-cell sequencing allows us to do is to actually go and measure each individual cell. And what that does in turn, it reveals all sorts of heterogeneity that may exist in your data. So it's a really cool technology. And so you can imagine that if I have a bulk Hi-C data, then I get this information about what regions are interacting in a bulk setting, so as a population average. But what would be really cool to have is to have this single-cell Hi-C data, which is now giving me information at a single-cell resolution. But here comes the problem. It turns out that generating these single-cell Hi-C data sets, it's extremely resource intensive. And so we came in with the question, what if we could build a computational model that could predict these single-cell Hi-C maps from some easy-to-acquire genomic measurement? And this is not a crazy hypothesis, because people have done this before. So in bulk setting, there are several studies that take these very easy-to-acquire genomic 1D signal, use machine learning or deep learning, and accurately predict bulk Hi-C maps. But then we are dealing with single-cell data, so it comes with its own set of challenges. The first one is we want to do supervised learning. That means we need an input and its corresponding output. So what we are looking for are single-cell data sets that have measured two measurements from the same individual cell. Luckily for us, these kind of things exist. And as we were thinking about this problem, two studies came out kind of close to each other. So the first study did this measurement where they measured methylation and single Hi-C from the same individual cell. And another study came out which had done the same thing, but this time they measured gene expression and Hi-C from the same individual cell. We picked the second study as our data set for training our model, just because single-cell gene expression is very widely available. So if we build a model that can predict Hi-C from gene expression, then it will be accessible to more researchers. So here is the general idea. We are going to take the single-cell gene expression, and we are going to feed it into our model, single cell, we call it SC graph Hi-C, and we are going to predict single cell Hi-C. But it's not that easy. So the first thing that we ran into was sparsity. So it turns out that single cell data sets are notoriously very sparse, be it gene expression, be it Hi-C. So we had to come up with a compromise, because when we were training the model on this individual single cell level, there was too much noise for the model to learn anything reasonable. So we came up with a compromise, which is we pseudo-bulked the data. And what that meant was we took these cell type specific clusters and we aggregated information from there. So the pros were that we were able to substantially reduce the noise, so our model was able to train, and at the same time, we still have cell type level heterogeneity in the predictions that we are making, which is still better than the bulk Hi-C predictions. The con is that, yes, we do lose individual single cell resolution here, but we decided to move on with this trade-off, given that it was very difficult to train the model at single cell resolution. The next challenge is that we are trying to predict chromatin organization or DNA organization from gene expression, and these two entities are very weakly correlated because they're a little bit far removed. So we had to bring in some structural reinforcement. So the first thing we did was we introduced a bulk Hi-C. And the idea here was that this bulk Hi-C would serve as the higher order structure that the model kind of learns on. And we will use the gene expression information to kind of deconvolute this bulk Hi-C into the pseudo-bulk single-cell Hi-C maps. We brought in some other information that was useful to the model as well. For example, CTCF motif scores. So these are just scores that represent how likely it is for the CTCF protein to bind. This is relevant because this protein mediates the 3D organization of the DNA. It's been used by previous studies, and this is not cell type specific. It's just calculated for the entire genome, so we brought that in. And along the same lines, we also had CPG scores that were available, and these scores correlate well with active and open regions, so this information would also be relevant for the model. So given all of this information, let me present you the actual model, SC graph Hi-C. As you can see, it's a pretty complicated architecture, but we'll walk through it slowly one by one. So first thing first are the inputs. So as I already mentioned, we have the pseudo-bulk gene expression data. We have the CTCF scores, and we have the CPG scores. We have them for each genomic region, so these will serve as our node features for the graph. And the graph itself would be created from the bulk Hi-C data that we have. So first thing first, putting together all the features. So we have the 3 genomic profiles. We concatenate them. Those are our features. And then we have this additional step of adding something called as a positional encoding. So first, I'm going to talk about what this is in the context of graphs, and then I'm going to talk about why we do this. So how do we calculate these positional encodings? Well, we take our graph that we built using our bulk Hi-C data, and we calculate what is known as a graph Laplacian. So this is just the degree matrix. Degree matrix is the number of edges of the node minus the adjacency matrix. And what the graph Laplacian captures is how smoothly is the information flowing over a graph function. What this means intuitively is that if my graph is very densely connected, then the change in the information would be small. It's smooth. Whereas if my graph is very disconnected, then that change in information would be, or the change in values would be drastic. So this is what the Laplacian does. We take its eigen decomposition, and these eigenvalues basically capture the underlying properties and structure of the graph, and we add it as our node features. Now the question is, why are we doing this? So if you are familiar at all with transformers that we use in large language models, the way the transformer architecture is built, it's going to treat each word in a sentence independent of each other, which is not ideal because the words in a sentence are actually connected or related to each other. So what we do is we add positional encodings to each word so that the transformer knows that the words that are closer to each other in the sentence are more likely to be related, and the ones that are far away are less likely to be related. So it's the exact same idea, but we are doing that for graphs here. So what we end up basically getting is we have extracted the features that are conditioned on their positions in the 3D space. So those are the features. We do some processing in the neural network to learn the local and the global relationships between these features. And we do that by first applying a 1D convolution. Again, just a weighted aggregation, very local. It's just capturing any close-by relationships that features may have. And then we have the self-attention layer of the encoder from the transformer. The idea here is that what I'm trying to learn is some notion of similarity between different parts of the input with respect to other parts of the input. So if I'm a part of the input and there's another part further away that is useful for, and is related to me, then I'll be able to learn that using the self-attention. So what we are doing here is basically learning a very rich node feature representation, which is combining both the local and the global relationships that may exist in these feature space. So our features are decided. Now we bring in our graph neural network. So we built the graph using the bulk IC. And here now we have the node features for all the nodes. Now we will do a bunch of graph convolution steps. And basically what we are doing is we are merging the bulk IC with the GNN operation and performing the deconvolution step. So this is the place where the model will learn to extract cell type specific structure that is informative for it to predict the last bit of predictions. And then last but not the least, we have a decoder module, and everything we are doing are these linear vectors. So we need to take that vector and transform it into a 2D matrix. So we do an inner product, feed it through a few more layers, and this is what creates the likelihood map, which is our pseudo-bulk single-cell Hi-C from the node encodings. So that's the entire architecture. Moving on to the experimental setup. So as I already mentioned, we have this high-res data set, which is a co-assay, gene expression, Hi-C from the same cell. They did this experiment on a bunch of different cell types for mouse embryonic development and brain cortex. For the training set, we kept chromosome 7 and 11 as testing and trained on everything else. These are all the different cell types that they have. We did keep 3 cell types completely separate as our held out test data. This is something that the model didn't see during training, and we tested the generalizability of the model on unseen cell types. For baselines, we didn't have any existing methods, so we basically did different variations of our model just to see which of our decision choices were relevant for it to make predictions. So what happens if it's only gene expression? What happened if we add CTCF, CPG, and then also a bulk-only baseline where we tested whether it was actually possible to deconvolute the bulk Hi-C without giving it the gene expression signal. Evaluation metrics, these are some similarity scores that the field of Hi-C uses, and it basically measures the similarity between 2 Hi-C maps. So we did that for our predicted pseudobulk single-cell Hi-C and the target one, and higher score is better. So looking at things qualitatively, we were able to generate some really nice pseudobulk single-cell Hi-C data sets, and here we have the target map over here. And what we found was that out of all the variations, the one that combined all our decision choices was the one that was really able to extract the finer scale features in the data. Analyzing this quantitatively, we noticed that adding the CTCF signal was the first boost that we got in our performance, suggesting that the model is really using it a lot for making predictions. And then the second boost we got was by doing this whole bulk Hi-C and deconvoluting its setup, which also supported our decision that we made. Looking at the held out cell types, so here the horizontal lines are the average performance on the training cell types and the predictions of the model on cell types that it hadn't seen, and the performance is roughly similar to what we had seen before. However, for the last column, the brain one, this entire time we were using the mouse embryonic stem cell bulk Hi-C as our prior. But once we switched it up with the cortex Hi-C prior, we saw a boost in performance, suggesting, unsurprisingly, that if you have a bulk Hi-C that's closer to the single cell data set that you are predicting, it'll be better for your predictions. So we also went ahead and did a more thorough analysis there. So we brought in bulk Hi-C for different developmental stages and fed it into our model just to see how the performance improved. And as expected, for bulk Hi-C data where there was very little structural prior for the model to work on, the performance was the lowest compared to the case where there was more structure in the bulk Hi-C data that the model could work with. And the final bit I wanted to present was a little bit more critical analysis of the predictions on the held out cell types. And this is where the prior bulk Hi-C information also came into play more. So we started looking map by map the areas or the regions where we expect to see cell type specific differences and how the model's predictions were there. So when the structure was preserved across the cell types, this was a nice case, trivial case for the model, and it does pretty well. For some cases where there is cell type specific differences between the Hi-C structure, the model was able to capture that, but not every single time. We did find cases where it was not able to detect the cell type specific structures, but for some of these cases, or a lot of these cases, this was resolved by integrating the bulk Hi-C that was closer to the single cell data set that we were working on. Again, really bringing home the point that the closer the bulk Hi-C prior is to the data set you are working with, the better it is. So with that, basically single cell graph Hi-C model, it predicts cell type specific pseudo-bulk contact maps for single cell Hi-C using pseudo-bulk gene expression in the single cell domain. We have shown that we can generalize to held out cell type samples, and we can replace the tissue-specific bulk Hi-C prior without the need for very fine tuning. And if it's closer to the data set that you are working with, the better performance you get. Our code is available if you're interested to test the model out, and this work was presented at ISMB last summer. Last but not the least, thanking all the people who worked on this project. So this project was led by my PhD student, Murtaza, who is graduating soon and heading to a postdoc position, along with our collaborator, Justin, and a very talented undergraduate student, Byron, in the lab. Big thanks to all the members of my lab. As you can see, we are a happy group of PhD and undergraduate students working on very critical challenges of multimodal data integration using machine learning. Big thanks to our funding sources for this project, as well as my affiliations, and I'm happy to take questions now.